Okay, good morning, everyone. This video is going to simply be a quick and hopefully not too painful introduction to some of the basic structure and some of the concepts that are going to underlie the class. This is Natural Resources 4120-6120, methods in population analysis or some such. And again, this is really intended only to provide you a little bit of an overview and a little bit of insight into the ideas that we're going to explore and a little bit about the mechanics and the structure. So we'll start by actually taking a step back and thinking a little bit about the larger motivations for the class. Many of you, or even most of you, have a background, at least at some level, in population dynamics. And those of you who've taken population dynamics with me will understand or recall that one of the things that I like to point out at the beginning of those sort of pop dynamics classes is that population dynamics is a fundamental base element of natural resources and conservation and sustainability. Population dynamics is a key to understanding lots of biology and a lot of the policy. Dynamics or dynamical systems, they're fundamental, and they're fundamental not just for ecological systems or natural systems, but they're also fundamental for chemistry and economics and physics and social dynamics and all sorts of stuff. And really the main sort of thrust that I'm trying to make here is that if you don't understand something about dynamics, it's really difficult to think in a kind of a rigorous way about things related to conservation and sustainability, since they rest on understanding changes in how many are there. And fundamentally with population dynamics, that's what we're after. And many of you will remember that the sort of basic equation for population dynamics, which is sometimes referred to as the BIDE equation, can be represented as we see here. So the number of individuals that we see in a discrete time step, like next time step, next year, for example, is a function of a bunch of things. So it's a function of how many do we have this year, which sort of makes intuitive sense. And then that mapping between where we are now, n sub t, and where we might be next year is going to be influenced by the number of births and immigrants and deaths and emigrants. So we have the additions, the births and the immigrants, and the subtractions, the deaths and the emigrants. And, BIDE, which is where the acronym comes from, reflects the basic processes which are going to influence the change potentially between where we are now and where we would be in a future time step. And so population dynamics is largely motivated by interests in changes in the population abundance over space or time. And what we see in this expression here, this sort of canonical equation relating the population size differences between two different points in time as a function of the underlying processes, survival, reproduction, and movement, immigration, emigration being sort of movement processes. And the key parameters, again, that are actually represented in that expression have to do with survival. How many of the individuals that are alive now are alive and available in the population the next time step? Movement, individuals moving in and out of populations, again, related to emigration and immigration processes. New individuals arising because of reproduction and recruitment. Recruitment, the probability that an individual born stays in the population. The size of the population, the rates of population size. So these are all sort of key parameters that we actually see in this BIDE equation, either explicitly or implicitly. And population dynamics is essentially the study then of either the sort of sub-processes, the lower level processes, or the overall impact of those processes on population size and how that changes through space and time. Now, when you kind of stare at the equation, the BIDE equation, so now we've simply moved a few things around. Delta N, the change in population, essentially is just the arithmetic difference between where we are next time step minus where we are now, is undeniably and simply algebraically a function of the birth and the immigration, the death and the immigration processes. So we can understand changes in the population size by looking at the underlying processes. And if you think about it for a moment, that's actually sort of the fundamental thing that we're we're likely to be interested in. Simply establishing that there's been change in abundance through space or time is one thing, but knowing what it is that has driven those changes is ultimately more important, because that's essentially what we're interested in terms of conservation sustainability. You don't manage abundance, you typically manage the processes that influence changes in abundance. Okay, so in theory, this sounds like a really simple thing to do, right? I mean, algebraically, it's simple. If you knew the births and the immigrants and the deaths and the immigrants, then you could talk intelligently and rigorously about what's going on the left-hand side. So simply count things. Count the number of births, count the number of deaths, count the number of immigrants and immigrants. It sounds relatively simple. Unfortunately, as we're going to determine, and this is essentially one of the major motivations for the class, in fact, it's not that simple. Okay. Simply consider just the left-hand side. Let's ignore for the moment the lower level processes, the B, I, and the D, and the E, and let's just look at the differences in abundance. Okay, that might even sound simpler. How many animals are there at two different points in time? Well, let's just take that question. How easy is that? All right, so how many are there? Well, so here's some caribou that we actually see, and the question is, well, how many are there? So some measure of population abundance. Okay, so the intuitive thing that you might sort of imagine is that this should be relatively straightforward. Okay, well, there's one. So here's our visual picture sample, and there's one. There's two, there's three. So, because we learned to count as sort of our first sort of quantitative skill, counting sort of integer values of things is pretty straightforward. But it's based upon this premise that you actually see three here in this picture. But how many are there? Well, 1, 2, 3, a lot. All right, so the ones that are forward in the picture you can count, but what about the ones in the back? Well, you see signs of the individuals, but you don't necessarily see the individuals. So how many are there? All right, and this is fundamentally one of the big problems. Okay, so even ignoring the underlying processes of births and immigration and death and emigration, simply establishing how many there are is difficult. And why is it difficult? What is the basic challenge for estimating abundance as an example type of problem that we're going to consider in the class? Well, the fundamental problem is that in most cases, you cannot conduct a census and therefore must sample the population of interest. Excuse me. So what do we mean by census? Well, in most cases, you're not actually counting everyone. You're looking at a sample of the population. All right. So a quick reminder, what do we mean by sampling? Well, sampling, one definition is the act or process or technique of selecting a representative, what happened there? Part of a population for the purpose of determining parameters or characteristics of the whole. All right. So if we look at this little diagram here, the target population in green is what we might be interested in estimating the abundance from. We can't, for a variety of reasons, maybe logistical or financial, we can't census the entire population. So we look at a sample, we take the piece of the population, we sample from it, and we're trying to establish from the sample, what can we say intelligently about the potential abundance of the entire population? Okay, so again, back to the fundamental problem. In most cases, you cannot conduct a true census, and therefore you're sampling the population of interest. So why is estimating abundance hard, or in fact, as is sort of indicated up here, any parameter? So here's the basic problem, okay? So you go out into a population and you count the number of individuals you find in your sample. All right, so let's say we're trying to enumerate the number of ladybugs. Okay, so you go out and you grab a sample of ladybugs and you count them. So then the question is, and the second one is sort of the key, is how many that you count a good measure of how big the population actually is? Right? And the answer is generally no. All right, so why? Well, the reason why is because you don't count everything. In very, very few circumstances do you count everything. Most of the time, 99% of the time, you don't. The number that you count is the number that you encounter. Okay? So look over here on the left-hand side. This is this diagram, this picture of an iceberg. So what's true about the basic properties of an iceberg? Well, here what we see with the iceberg, above the water, we see some of the iceberg, but the vast majority of the iceberg is actually below the water. So why use this picture? Well, this picture is actually a perfect analogy of the challenge with sampling. You go out into a population, and what you encounter, what you see, like here in the iceberg above the water, is only a representation of the whole iceberg. Okay, so this is the observed part, and the total size of the iceberg, in fact, is the observed part plus the unobserved part. Okay, so go back to the context of population sampling. You typically don't encounter all the individuals, just like you don't see all of the iceberg. So if you miss or don't encounter some of the individuals, then the number that you encountered will be lower or smaller than the number that are actually there. If you try to estimate the size of the iceberg just based on what you see, you're going to be very much negatively biased. It's going to be way too small because clearly the amount that you see is a lot less than the total size of the iceberg. This is the same as using the count of the number of individuals as a measure of the number of individuals in a population. Okay, so we're going to demonstrate that by means of a simple example, okay, a little simple kind of problem that I want you to work on. So on the next slide, you're going to see a sample of some text. It's some text actually taken from the text of the Peter and the Wolf story. So in this piece of text, there are a number of typographical or spelling or other sorts of errors in the text. So you're going to pretend that you're a proofreader, right? And this is actually a real world problem. Proofreaders try to determine how many errors there are. And then when the number of errors gets below a certain value, they think it might be reasonable to go ahead and publish the book. Okay, so how many errors are there is essentially a problem in how many individuals are there. So in the piece of text that you're going to see in a moment, there are a fixed number of errors. So the population size is fixed. And so I'm going to give you 60 seconds to actually try to determine how many errors that you see. Okay, so here in the video, what I'd like you to do when I bring up the slide is pause the video for 60 seconds or so and see how many of the errors you can actually count. And then we're going to look to see how many there are, and then we're going to talk a little bit about why you probably didn't actually count all of the typographical errors and the other sorts of errors in the text. All right, so get ready with a piece of paper and see if you can write down the number of errors. Here we go. Okay, so let's stop here and let's actually go and see how many errors that you actually have. So in fact, there are 17 errors. It's a fixed number of errors. And how many did you detect? So the vast majority of you are going to get fewer than 17 errors. Hopefully nobody gets more than 17 errors, but that can happen too. And we're going to talk about that a little bit. Okay, so here are the errors, okay? So there's 17 of them. If you simply count the things that are in red, you'll see that there are 17 total errors. Some of them are, just spelling errors. Some of them are kind of weird errors that have accents that shouldn't be there. Some of them are grammatical errors. Some of them are punctuation errors. So if you look at this for a few moments, you'll see what each of the errors represents. There's 17 of them. Now, once you've looked at this for a moment, and if you go back and look, what errors did you see and what errors didn't you see? So some of these errors are very subtle. Some of these errors are very obvious, okay? So this is not the way you spell alarmingly, and this is not the way that you spell frantically, okay? And here the word should be safety, not safely, and that's not how you spell tree. But, occasionally the errors are subtle, like the sort of reversed and inverted E, double quote here when it should have been a single quote, or at least the quote should have been consistent, and so on and so forth. So there's a fixed number of errors. Not all errors are equally detectable. And that's the sort of point here. So when you go back to this, we have several things that are actually happening here. All right. We have a certain fixed number of errors. Some of them you're going to detect, some of them you didn't detect. But the main point is that the number that you probably counted is lower than the total number that there actually are. You probably ended up with a value that was less than 17. Some of the reasons are related to this fact that some of the individual errors are less detectable than others. This is going to be true in population studies as well. Bigger animals may be more detectable than small animals. Brightly colored animals may be more detectable than less brightly colored animals, and so forth. So this is, again, the part of the basic ideas that we're going to develop in the course, having to deal with the fact that there may be a certain number of animals, but you don't detect all of them typically within some sort of sampling time frame. So I gave you 60 seconds. Maybe if I'd given you 5 minutes, you would have found them all, but I gave you 60 seconds. And so in 60 seconds, you didn't detect them all. Okay. So what does that do for us? What do we actually do with that basic idea that you don't detect everybody? Well, what we want to do is quantitatively take into account that you don't necessarily detect everyone. And so we're going to introduce a parameter P, which is the probability of seeing or detecting the animal if it in fact is there. So in the context of the typographical error problem, it's the probability of detecting the error if it's there. Well, the errors are there. They were put in and they're not moving around. They're not immigrating or emigrating. It's fixed. So P is the probability of detecting the individual. Okay. And so P is a probability. So if the probability of seeing everybody, all the errors in the example was 1, then that's fine. We're done. Basically, then there's a count. But it's not, because there's some probability that you're going to miss the animal or miss the error in the typographical proofreading experiment. So if P is the probability of seeing the animal, then 1 minus P is the probability that you miss it, that you don't detect it, even though it is really there. Okay, and that's the key. So P is the probability of detecting the animal if it's there, and 1 minus P is the probability that you miss it, even though it's really there. Okay, so now let's put those two things together. How many do you actually count? Well, how many you actually count in your survey is a product of two things. It's the product of how many do you actually have times the probability of detecting the error, or detecting the animal given that it's there. Okay, so let's look at this a little bit. So if the number that you count during your survey is C, and we accept algebraically that it's the number that are really there times the probability of detecting them, then by simple algebra, we could rearrange things and come up with an estimator for N. We could simply take our count and divide it by or correct it for the detection probability. So if you just look at the algebra of this first expression, we're just rearranging it in terms of N. And so it's simply the count divided by the detection probability. So the count is your minimum population size. You know, if you count 11 errors, well, you know it can't be any less than that. But we now need to correct it for the detection probability. The count will only equal N if P is 1, and that almost never happens. So the count divided by the detection probability will give you N. So let's consider a really sort of simple, trivial example of this. Suppose you go out and you count 50 individuals during some sample of some population. So the count is 50. Suppose that we know that the probability of the encounter of an individual, given that it's really there, is 0.25. Well, again, what do we mean by 0.25? That means there's a 1 in 4 chance that you'll encounter the animal or detect it. All right. So you're wandering through your forest or the field or wherever you're doing your study. Oops. And there's a 1 in 4 chance that the animal who's actually there, that you're going to detect it. Okay. So given those two pieces of information, then your estimate of the abundance is simply the count 50 divided by the detection probability 0.25, which equals 200. All right, now just to sort of reinforce the logic, suppose that the true population size is 200, but there's only a 25% chance that you're going to detect any of them. Okay, that's what 0.25 means. That's what P equals 0.25 means. Then what's 0.25 of 200? Well, it's 50. So there's 200 individuals. There's a 25% chance that you'll detect any one of them, which means the expected count should be 50. All right, so that's all the numbers sort of hang together. Now, of course, in the preceding, we assumed that we knew P, but what if you don't? And that's essentially what the course is largely about. The estimation of abundance or density. And as it turns out, almost every parameter that we're interested in is generally an exercise in estimating P, which we use to correct the count. So the estimator of N, it's got a little hat over it, meaning we're talking about a statistical estimator, is the count, which is not an estimate. It's actually an empirical measure of something. You counted a certain number of animals divided by the estimate of P. So a lot of what we're going to focus on in this course is how do you estimate the detection probability? And the key step is usually based upon what we refer to as multiple encounters. What do we mean by multiple encounters? Multiple encounters means you grab an individual and you mark it, let's say in some fashion, or you write down some individual characteristic of the animal, you release it, and you go back out and you look for it again. And if you detect the animal again, that's a multiple encounter. Okay, if you only detect it once, that's a single encounter. And from a single encounter, there's a limit to what we can do. But if you go out and you release an animal and you look for it multiple occasions and you actually do detect it multiple times, that's the basis of the information that we're going to need. And we'll see exactly how all that works when we get into the class. So in animal population sampling or monitoring, what we're looking for is the data that we're going to need to estimate detectability. All right. And the key to doing this, and the key idea to always keep in mind, is that even in the areas that you're surveying, you don't detect everybody. So we need a solution for estimating P, the probability of detecting those animals, and that's based upon repeat encounters. Okay. So what constitutes encounter data? Well, there's lots and lots of different sources of encounter data, and this has evolved and expanded over the years. One of the traditional things was you simply physically recapture animals. So here we have a robin and a mist net. On the initial encounter, you put some kind of leg marker on it so that you can identify that individual in the future. And then conditional upon it being alive and in the sample, you go back out and you see if you catch it again. So if you catch it more than once, then it's multiple encounter data. These days we can do it not necessarily by physically marking the animal, by looking at the DNA of the animal under the assumption that the DNA is a unique individual marker for that animal. Sometimes we don't want to physically capture them, or we can't physically capture them, so we use markers that we can observe from a distance. So here for this snow goose, we have a neck collar on that individual. Telemetry, you put a radio transmitting device on the animals, and you may not physically capture them, you may not even see them, but the device that you're using will detect their radio signal. And we can also use indications sometimes from the animal's characteristics. One of the very common things that cetacean biologists do is they look for characteristic scarring patterns on whales or dolphins. A lot of animals, walruses, all have these sort of marks which sometimes can be used to identify them from a distance. And for non-water things, this is also very commonly used for tigers and jaguars and a lot of big cats that have sort of unique coloring or unique whisker patterns, which sometimes you can observe from a distance. And increasingly, in a nod to the folks at the Lab of Ornithology, we can use the sounds that individual animals make. Each individual animal has a fairly characteristic sonogram that you can often use to help identify the number of individuals that you may be looking for. Okay, so key classes of encounter history data that you might collect by the methods that we talked about. We sort of broadly distinguish between live encounters or live captures where the animal is alive. But there's also data that you can collect from dead recoveries. So for things that are harvested, either for recreational purposes or for commercial purposes, you have information, okay? You know the information is based upon the harvested individual. And increasingly, we can also do things that are simply related to the general question at a species level of whether species are simply detected or not. Right? We're going to talk a lot about all of these different classes of encounter history data and how you use them over the course of the semester. Now, another thing which has emerged recently, I'll say within the last 10 years, is this idea that we may be able to make reasonably defensible inference simply by counting the number of individuals that we detect at any particular point in time. And what we have here on the right is representation of that. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 of these goldfish in our sample. And if we were to make multiple samples over space and time, and we had a good rigorous count of how many did we detect, then depending upon a bunch of statistical assumptions you can make, you can actually tell a reasonably defensible story about population size or population dynamics if the assumptions are met. And the key assumptions that you're making is that you don't count the same individuals more than once. But if the individuals aren't marked, how do you know that for sure? So under some fairly strong statistical assumptions, you can use unmarked counts of things to tell stories. But again, you have to really rely heavily on fairly strong statistical assumptions. Now, there's an awful lot of data that fall into that category of unmarked counts. So there's been a huge amount of work done in the past 10 years or so on what you can do with those data. But I don't think anyone would dispute that the gold standard is what we have over here on the left, where we have individually marked organisms. Okay, so if you have individuals that you can follow through time, then that's the best of all worlds in terms of being able to tell a story about changes in abundance and the dynamics of populations through time. So for this class, we're not going to talk at all about the unmarked stuff that you can do. We may get to a little bit of that later in the semester, but the vast majority of time, we're going to focus in on what you can do with marked individuals. And these key parameters, which we introduced earlier, as all being sort of fundamental components of population dynamics. If you have marked individuals, all of these are estimable, statistically estimable within a rigorous framework. Okay, and we're going to look at all of these things over the course of this semester. So the modern methods which have evolved over the past 40 or 50 years will allow you to estimate all those parameters, including abundance. And what's I think is really interesting and worth keeping in mind is we're going to focus on sort of populations and animals and living organisms, but you can apply these methods generally whenever you want to account for the probability of an event which may not be seen. Right? So as we get through the course of the semester, we'll actually look at a few examples of where that can be done. And in fact, we introduced an example earlier. The use of some of the technologies that we talk about in the course is exactly what professional publishing companies use to estimate the number of errors in the text for books that they're publishing. It's also applied for estimating the number of errors in software code. There's all sorts of ways that we can apply the technologies that we're going to talk about in this class to things beyond sort of population ecology. Now, this last point here is an important one. It's an area of intense research. And by intense, I do mean intense and prolific. There are probably 125 to 150 papers being published per year. And there are very, very few people who can possibly keep up with the full span of the literature. Now, the good news is, I guess there's two levels of good news. One good piece of good news is I don't expect you to do any more in terms of trying to read everything than I do or any of us do. It's not possible. Nobody could read everything that comes out. But a huge proportion of the major advances show up in the software fairly quickly. So keeping abreast of what you can do in the software is often a really convenient way to keep abreast of the literature. So the software often acts as a filter and a collation for all of the new things that have come out. And so that's kind of the approach that we're going to take. We're not going to spend a huge amount of time for the majority of the course reading much of any of the primary literature. We're going to accept that the software collates a lot of that for us. And so we're not going to spend time trying to read a lot of journal articles. So what are we actually going to do then? Well, we're going to sort of split the course into sort of two pieces. I mean, administratively, there's this sort of de facto idea of a lecture versus a lab. The distinction is pretty fuzzy for this class, but we are going to lecture to provide background theory and model structures and sort of the thinking about how it all works. And then we're going to actually figure out how to do it on a computer. So lectures and labs will largely be interdigitated. There will be some tendency to use the Friday lab period, which is longer, to do more of the hands-on stuff, but it's not going to be a strict separation between lectures and labs. But there will be an awful lot of focus on doing the two things. Now, that focus is going to be sort of clearly laid out for you in the syllabus, which you have and is also available on the course website. And the details for getting on the course website are on the first page of the syllabus. The primary piece of software that we're going to use, at least for the first sort of 2/3, 3/4 of the course, is a piece of software called Mark, program Mark. And again, just thinking back to a comment I made a moment ago, an awful lot of the new ideas and thinking is put into program Mark. So by learning how Mark works and learning to use Mark, you're going to understand an awful lot of things. And to sort of aid and abet that, again, rather than reading the primary literature, we're going to make a fairly heavy use of what's called the Mark book. It's a book that I wrote along with Gary White. Gary White is the guy who wrote the software, and I basically wrote the book about how to use the software. So Gary and I kind of tag team on this, and there are a lot of other of our colleagues who've made contributions over the years. And it's required reading. It's not required to purchase it, but it is required reading. And in all of the chapters of the book are available online for free, so you don't have to pay for anything if you don't want to. Most students find it convenient to purchase hard copy because most of us are more comfortable having something on our desk that's printed. We can look at that, and then we can look at our computer and kind of go back and forth. But that's entirely up to you. The reading isn't up to you. That's going to be what's required, but the purchase of a physical hard copy is entirely up to you. Additional readings and lectures and other bits of assignment, data files and all that stuff will be posted to the course website, and links to both the software, including, Mark, and anything else that we use will also be found on the course website. Now, each of the sort of main topics is going to follow a particular kind of teaching sequence. We're going to start by motivating the model. What do you want to estimate? Okay, so do you want to estimate abundance? Do you want to estimate survival? But if all we do is teach you how to do that, then we've missed, I think, a very important piece. And that important piece is why. When I have a student come into my office to say, I want to estimate abundance, I usually don't start by saying, well, show me your data and let's figure out how to do it. I usually start by asking the question, well, why do you want to estimate abundance? What scientifically is abundance of interest for? And then we start talking about, well, what kind of data types would you need to be able to estimate that particular parameter? Okay, so motivation in thinking about what you need and why is a fundamental first step, and we're absolutely going to do that. And then we get into the theory. How do you go from the questions like, well, I want to estimate abundance and here's why. Well, what models, what kind of data, what kind of statistical tools do I need? And what are the assumptions? What are the conditions and assumptions that we need in order to be able to come up with a rigorous estimate? And then only after we've gone for these first two steps are we going to get into the mechanics. How do you do this in Mark? And then we'll spend a lot of time doing hands-on examples. Now, if you look at the syllabus, the first 2/3, maybe 3/4 of the course is going to be focused on using program Mark. And that's fine, except that there are some new things that can't be done easily in Mark. And increasingly, people are choosing to use different sort of compute environments to do a lot of their analytical work. So we're also going to talk about how you can do some of the interesting things in R. And again, we'll follow the exact same thing. What are the motivations of the models? What do you want to estimate and why? What's the basic theory? Let's get down into the weeds a little bit about the statistical models. And then how do you do it? Right? So, in practice, most of us use combinations of various different pieces of software. So we're going to start by using Mark, because once you fully understand how Mark works, then it becomes a lot simpler to figure out how to do things, often more efficiently using R. But if you start with R, it tends to get very confusing very quickly, because R makes fairly significant presumptions about your level of understanding. And if you go through Mark first, you're going to understand things at a much, much higher level before you get into some of the nuances and some of the challenges of doing things in R. Okay. Now, there are, again, two key components of this class, and I would like you to always keep this in mind. We need to understand the theory behind what we're doing, both the sort of motivational theory about what parameters we're interested in and why, but also the statistical theory. And we also want to talk about the mechanics. If all we did was theory, then we don't know how to do anything and we're frustrated. On the other hand, if all we know how to do is run a piece of software without understanding the theory, then eventually you're going to run into a wall. You're not going to be able to actually be creative or really particularly intelligent about applying the software to particular problems. So we want to put the two things together. And that's essentially what we're going to try to do in this course. We're going to look at the two questions together of why and how. Why being the theory and how being the mechanics. Okay? And so at various points in the course, we're going to make it, really clear that we need to understand not just the mechanics, but also the theory. Now, the overall approach and objectives, I guess, sort of the learning objectives, is, despite the title of the course, which I think has wildlife in it for sort of historical reasons, it's taxoneutral. We're going to talk about, animals, we're going to talk about plants, we'll talk about all sorts of different things, but it's largely taxon-neutral. The main assumption that we're going to make going in is that you have minimal background both with the specifics of using the software, but I'm also going to assume minimal background statistically. Okay. And for some of you who do have a strong statistical background, hopefully that won't be frustrating. In fact, it might be a good opportunity for you to sort of learn some of the kind of key fundamental things. Most people that I know with decent statistical backgrounds often have reach the point where they understand how to do things better than they understand why. And so this is a chance to go back and learn why. But even if your statistical background is limited to the standard single semester undergraduate class, you're going to learn a lot. Okay. And you're going to learn a lot from an intuitive fundamental thing with minimum sort of reliance on anything mathematical. Okay. I want to stress familiarity with larger concepts. And we're going to develop some familiarity with important designs, both analytical and sampling. So one of the outcomes of the class that really wasn't intended when I started doing this many, 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 many years ago was that you're going to learn not just how to estimate population parameters, which is largely, a main motivation of the class, but you're going to understand a whole lot more about statistics and modeling in general. So for those of you who've taken other classes where we've used models, the motivation for doing that is something that we've talked about, but here in this class, we're going to talk about the statistical estimation of the parameters for those models. And in the process of doing that, we're going to understand a little bit more about statistics. Okay, so you're going to be exposed to some of the available software. I mean, Program Mark is leading edge. It's the de facto standard, okay, for this kind of analysis. And so you're going to be exposed to the same software that professionals use all over the world. And I want you to ask questions. We want to make this as interactive as possible. If you're unsure about something, then you need to ask the questions, okay? Now, this year, for the first time, I'm going to make this class a team effort in terms of the teaching. And there's a variety of reasons for this, but the main reason is that we now have a very sort of large group of people who are expert in a lot of the stuff that we're going to do in this course and have particular expertise in some of the really new and cutting edge stuff. And so if you look on the syllabus, you'll see at the top people are listed, and here they are. So we got Viviana Ruiz Gutierrez, Courtney Davis, Orrin Robinson, and Andrew Stillman. And these are 4 folks that are working at the Lab of Ornithology. Some of them I've known for a long time. Viviana, I was on her dissertation committee. I won't say many, many years ago, because then she'll accuse me of saying that she's really old. She's not. I've just known Viviana for a while. And she started learning some of this material with me back as part of her dissertation work. Courtney Davis was an undergraduate who took this very class a few years ago. Orrin Robinson was a postdoc that Viviana and I recruited not too many years ago. And Andrew is a recent hire at the Lab of Ornithology. And these four folks are doing some really, amazing work kind of on the cutting edge of some of the things that we can do. And I thought it would be both appropriate for them to do some of the instruction, since they're actually working in areas that I'm aware of, but I don't spend as much of my time on as they do. But also, this is the vanguard of the folks that are taking leadership positions moving forward. And so I think it's sort of important professionally for everybody to get to know the players, and these four individuals are the players. Okay, so what these guys are going to focus in on, and if you look at the syllabus or things sort of in the last third of the course, they're going to make sort of use of R. Again, I mentioned this a little bit earlier. You can do an awful lot of the analyses that we're going to talk about this semester using R, or at least R as a front end to mark. But I'm going to leave it to these folks to introduce the basics of using R. We're going to focus in on a particular class of models, at least to begin with, called occupancy models. For those of you who took 3100 with me, this is essentially the type of modeling that you do when you're dealing with the proportion of patches that are occupied. Some of you will remember the metapopulation stuff that we did in that particular class, 3100, where we talked about rates of change in the proportion occupied. Well, the statistical modeling of proportions occupied is largely referred to as an occupancy model. And that's going to be covered by these folks near the end of the semester, the last sort of third of the term. And then we're also going to talk about integrated population models. And the integrated population models is essentially one approach that's been proposed to solve this, I don't want to call it a dichotomy, but this sort of differential use of marked individuals versus unmarked individuals. You'll recall from an earlier slide that there are these sort of two different data streams that you can use, some where you have individuals that are marked or some where you don't. And recently there's been some attempt to actually integrate the two together, which not only do they sort of benefit each other, but it helps mitigate or at least minimize the number of sort of heroic assumptions that you need to make sometimes, especially with respect to the unmarked individuals. So we're going to talk a little bit about that as well. Okay, so this is essentially just the introduction. We're going to start for real on Thursday. And so between now and not this Thursday, but now and the end of the sort of second week, we're going to need to make sure that everybody's compute environment is set up and that everybody has whatever materials that we think you're going to need, and we'll be ready to start. So the first week, maybe 5 lectures or so, is going to be conceptual. There'll be some computer work, just to sort of reinforce some concepts, but we're going to start with program mark next week at some point. So no huge concern to get all of that in place to begin with. Okay, so that's it for now. And the first sort of main fundamental lecture will be lecture 1, where we talk about one of the main sort of frameworks for estimating parameters based upon the kinds of data that we're typically faced with, and that's maximum likelihood estimation. And that'll be next lecture. Okay, take care.